So welcome to our session on the use of acoustic telemetry for monitoring the effects of offshore wind development along the U.S. East Coast. I'm Chris Sarro. I'll be moderating this session with my colleague, Greg DeCelles. So just general format of how this will work. I'll provide a general introduction. That'll be followed by presentations from the individual researchers. Q&A session will be moderated by Greg. And just a reminder to please submit your questions through the app. So our speakers today, again, myself and Greg, we're from Ørsted, Connor Capanzano from Inspire Environmental, Allison Frey from the School of Marine Science and Technology, Michael Frisk from Stony Brook University, Bradley Peterson, also from Stony Brook University, Matthew Sclafani from Cornell Cooperative Extension, and Keith Dunton from Monmouth University. So just in the interest of time, I'm going to provide a high-level overview of the basics of acoustic telemetry. Acoustic transmitters or tags are implanted or attached to individuals. Here you can see researchers from the New England Aquarium putting a tag in the abdominal cavity of a bluefin tuna, and those animals are then released. Now, each tag will emit a unique signal at a predetermined interval, so every 30 seconds, every minute, every hour, that tag will emit its signal. So as the animals are moving around their environment and the tag is pinging, when they get close enough within range of an acoustic receiver, that receiver picks up the tag and any associated data with it and stores that information on the tag so it can then be downloaded later. Now, tags can be detected by any compatible receiver, which really adds power to acoustic telemetry. So if you tag an animal up here off of New York and it swims past a compatible receiver off of Florida, that information is stored. And regional data sharing networks really allow researchers to track animals across a much broader spatial scale than their own receiver array that they might maintain. Now, just a little bit on array designs. It can depend on any number of factors, but two very important ones, are your research questions and the target species that you're looking at. So if you're just looking at, say, movement in and out of an estuary, you can set up a gate of receivers, the mouth of that estuary, and that will allow you to keep track of what's going in and what's going out. Generally speaking, arrays used to examine fine-scale movement require a higher concentration of receivers in a given area than an array that might be used to examine just presence-absence, general movement, and or residency. Those arrays can be much more spread out. So an example of this is actually the array that Dr. Frisk put out on the south side of Long Island. The receivers that are concentrated in the middle are used to track fine-scale movement, and the receivers along the outside are there for just more general information. The behavior of the animal also comes into play here. If you're looking at an animal that has a small home range, you don't need to spread receivers out for miles because they don't really travel that far. That conversely, if you're looking at a species like tuna or shark, Connor will get into more detail about this, but you want to spread your receiver array out further because the animals just cover a lot more ground, so you want to be able to pick them up as they move through the environment. So again, that's just a high-level overview. I'm now going to turn it over to the individual speakers. Connor is up first, and he's going to be talking about the highly migratory species work that is being done in southern New England.
All right, thank you, Chris. Thank you, everyone, for joining us. Again, my name is Connor Capanzano. I'm a fisheries scientist at Inspire Environmental, and I am not Jeff Kneebone, and I am not Brian DeCelles. I'm speaking on their behalf today because they're in the field enjoying themselves. So I'm going to be talking about, as we mentioned, the work that they have been doing, such as creating and maintaining a large-scale acoustic telemetry array off of southern New England. Now, in the interest of time, I'm going to be giving a high-level overview in regards to how the project came to be, how it's evolved, some of the challenges, and finally some preliminary data that we've been able to actually look at. Now, this project, as Chris had identified, is primarily focused on highly migratory species. These are your pelagic fishes, your sharks, tunas, and billfish. And this particular project, we all know southern New England is important, of course, to offshore wind energy development. But in regards to HMS, there are some close relationships. One of which is that southern New England contains essential fish habitat for multiple highly migratory species for migration, foraging areas, and to satisfy other life history needs. Additionally, Southern New England supports a large recreational fishery for highly migratory species, some of which target bluefin tuna, blue shark, and shortfin mako. These particular figures are looking at federal surveys. The top row is the large pelagic survey, which is an intercept survey at the dock, getting general locations where anglers are going out to catch HMS. And then the bottom row is looking at conventional tagging data from the federal government in regards to where fish are caught and tagged with conventional tags. You can see here that based on some of the trends, both inside and outside, there is a high degree of effort surrounding this particular area. So to this end, the co-PIs submitted a proposal in 2019 to the Massachusetts Clean Energy Center to try to establish an acoustic array to monitor HMS off this region. Now, again, they were going to get a small number of receivers, so the basic questions they had were very simple in regards to focusing on some primary species of the fishery, those being the ones I've mentioned previously, bluefin tuna, blue shark, and shortfin mako, and then answering, again, basic questions. Are they in the area? Are they at popular fishing locations? Are they in lease areas? And if so, how often and what do the movements look like? So in 2020, they received funding for that 2019 proposal. And on the left, they started off with 16 acoustic receivers that were primarily concentrated in the northwestern portion of the wind energy area. And then over a span of 4 to 5 years, in 2024, we get the final product that we're looking at now. This timeline on the bottom gives you a rough idea of how many receivers were added, which somehow changed in their orientation. So that says 16, 38, 80, 77, 71. Glad we're on the same page. So showing just how, as lease areas opened up and they had more developers come on board, we were able to increase the number of receivers in these offshore areas. And obviously, we also have these gray blocks and red blocks, which identify deployment durations for receivers. Now, in New England, especially in the winter months, people like to bring receivers in because of it's tough to get out there to recover information, especially to maintain them. And then finally, if you lose a receiver, you lose all of the data leading up until that point. So you can see starting off at roughly 5 months, then over time, these receivers are out for 7 months at a time. And then we had some challenges. Some of those challenges include where to position these receivers in lease areas to maximize detection likelihood, right? And in some cases, these receivers were actually moved to areas where receivers were taken out due to the co-occurrence of other tagging projects like Ali will talk about in a few minutes. So receivers were constantly moved to make sure they're maximizing detection. But one of the biggest challenges was receiver loss. Right, we have an active fishery in southern New England, trawl fisheries, and we had receivers being snagged and losing those data. So the co-PIs worked with fishing industry personnel to identify specific areas that the industry partners don't frequent in fear of perhaps snagging their gear on obstructions or other types of wrecks called hangs. So the group started repositioning their receivers on these hangs and providing specific coordinates to the fishing community so they could avoid them. And in doing so, in recent years, they've had very little receiver loss. Now, another challenge with this particular project was data sharing, right? Up until this point, I should say 2021, all the work was being done on a lease-by-lease basis, right? Collecting data in a lease, reporting data to said leaseholder. And it's really difficult to get regional insights without having all those data in one data set. So in 2021, the co-PIs reached out to the developers requesting that these data could be aggregated for a regional assessment. They were successful in doing so. That involved efforts from 5 developers, 6 contracts, and 8 lease areas, and they were able to look at data from up to 80 receivers and currently 258 transmitters. Now, I'd like to point out that this particular data sharing agreement does not include the broader data sharing network, such as the ACT network or Matos. Not all developers were unanimous in the choice to distribute that information. So there is a need for continued talks to understand what data can be shared and what cannot, and also what format, in raw form or aggregated for confidentiality's sake. So now we're going to get into more of the fun part, which is some of the data that we have over the entire wind energy area. As I mentioned, roughly 258 tags have been deployed through 2023. Each point references a location where a tag was deployed with that specific animal. Shapes identify the year that happened, and then colors correspond to particular species. Now, as I mentioned, there were 3 main species, but over time and opportunistically, they tagged other species such as dusky sharks and yellowfin tuna. And you may notice some patterning in regards to where fish were released in a given year. And that's just based on a recreational fishing effort and where the fish were at that time of year. Now, everyone, hold on. This is not as daunting as it looks. Take a breath, hold on to your chair. So just starting off in the top left corner, quick breakdown of the top 5 species that were tagged of the 258, including blue sharks, shortfin mako, dusky, bluefin tuna, and yellowfin. And basically, of the animals that were tagged, roughly 78% of them were detected in that larger array area. When looking at the times that they were in the wind energy area, they varied from 1 minute to about 50 hours, and there were species-specific differences in those averages, ranging from 40 to 80 minutes. Some fun tidbits: some blue sharks were detected 4 years straight, and other species came back for several years after as well. So this particular figure is looking at detection history for a particular species by color, and then on the y-axis we have receiver over time for 2023. And what I want to point out is these vertical lines. These vertical lines represent the first time a tag was put out in 2023 for a given species. So looking at orange, which is shortfin mako, anything after July 1st is when tagging efforts started for that year. So you'll notice there's a larger increase in tag detections for makos as more tags went out, and they resided in the area. Anything before that indicate that fish were returning to the area. So just giving people an idea that we had fish coming back to the area, and then when they were here, they stayed for most of the summer into the late fall. Now, why is this all important? Obviously, and this visualization will help demonstrate that. Regional monitoring, when we have all these data together, allow us to look at the holistic view of what's going on in the area and identify linkages between the wind energy area, both inside and maybe where they're going through. This is not truly possible with a lease-by-lease application. So again, we need these regional monitoring efforts to help inform marine spatial planning activities. So to date, the co-PIs are going to continue with their multi-lease assessment and also enhance the way they continue with analyses, their monitoring efforts in 2026 in their field applications. Overall, they're going to continue to try to achieve full data sharing capabilities and push towards larger regional assessments and also tagging other species of recreational and commercial importance. And with that, I hand it over and thank everyone who was involved in this project.
All right, hi everyone. My name is Allie Frey, and I'm a PhD student and research technician at UMass Dartmouth School for Marine Science and Technology. And today I'll be discussing some of my dissertation work where we are using acoustic telemetry to assess interactions between offshore wind development and cod spawning on cocksledge. So just a brief overview of this talk. First, I'm going to talk a little bit about why we're doing this work, our motivation and objectives, and then some of the work we've been doing during the pre-construction phase of offshore wind development, as well as our plans for a post-construction assessment, and then some general conclusions. So just to orient everyone, our study site is in southern New England, in the southern New England block of offshore wind development areas. And we are focusing on cod. Cod support a recreational for-hire fishery in this area. They've been historically important, and their population levels are not doing great. Cod in southern New England are a newly identified stock area and have been going through a stock assessment process. So it's a pretty important time to kind of focus in on this group of cod and figure out information about their spawning dynamics and their population. So cocksledge is an area basically right by the South Fork lease area, which is kind of in that lighter green color. You can see Revolution to the north in the orange. Both of these areas overlap with cocksledge, which is an area with complex benthic habitat and is a well-known historic spawning ground for codfish. So these areas overlap with designated essential fish habitat, as well as the recently established habitat area of particular concern. And so we're really trying to figure out how cod in this area may be impacted by the development of offshore wind farms. Cod spawning aggregations are sensitive to human disturbance. Cod form dense groups with complex behavior. They produce sounds which could be masked by the sound produced by offshore wind operations, and have been shown to be sensitive to human disturbance. For example, interactions with fisheries in the Gulf of Maine were shown to interfere with cod spawning aggregations. There have been several studies in Europe which have looked at cod behavior around and within offshore wind farms. A lot of them showed positive impacts, with cod benefiting from the increased structure in the water, a higher prevalence of prey, as well as structure and shelter. However, as we've been discussing throughout this conference, the habitats in the North Sea are different, the fisheries are different, the management is different. So we really need a region-specific study in southern New England to really tease out what would happen when we build a wind farm on a cod spawning ground. So we had three main objectives. First, to broaden our knowledge of southern New England cod biology and spawning dynamics. Second, to collect pre-construction baseline data of habitat use and spawning dynamics. And then third, we plan to collect post-construction data to address potential effects of offshore wind energy development. So our pre-construction phase of the project started in November 2019 and continued through May of 2023, right before construction began. We used primarily acoustic telemetry methods, as well as some passive acoustic monitoring, but I won't be discussing that in this talk. We worked very cooperatively with Rhode Island commercial and for-hire recreational fishermen to kind of develop our study plan where we were going to monitor certain hot spots for fishing and cod activity. And so we developed the array, which you can see on the right side. The orange highlight is the South Fork area, just to orient you. The red points are our bottom-mounted fixed telemetry receivers. And then the vertical lines are a glider track. We had one receiver mounted on an autonomous glider to survey a much broader region. So the bottom-mounted receivers were left in year-round to really try to capture the arrival and dispersal times of cod to this area. The glider was deployed during winter spawning months, and you can see a picture of the glider there, as well as the bottom-mounted receiver. This pre-construction phase was done heavily in collaboration with scientists from the Massachusetts Division of Marine Fisheries and the NOAA Northeast Fisheries Science Center, as well as other groups. So here's kind of our process. We were targeting large reproductively mature cod, tagging them with VEMCO V16 transmitters. We deployed 100 tags over 3 years, and we also conducted fisheries-dependent and independent sampling to observe maturity status of these fish, which helped us bolster our assumption that the fish we were tagging were in spawning condition. And here's just a quick map of our releases. The yellow points are locations of where we tagged fish, and you can see most of them were kind of right within the array. That's not by coincidence. We had the array there because those are fishing hotspots. And so a quick look at some of our results. These are 4 of the winter spawning seasons. So from November to April, the data was filtered for any potential mortality. And you can see our fixed bottom-mounted receivers are scaled by number of detections. So the larger the circle, the more detections we saw there. And then the blue triangles represent telemetry detections by our glider. The squiggly line is the glider path, the actual path that the glider followed. So when we pull it all together, you can really see three primary areas where we saw a lot of detection activity, slightly to the northeast of the South Fork area, to the southwest, and then a little bit to the west as well. So looking at the temporal pattern of our pre-construction results, just to orient you, we have each individual codfish on the y-axis and then time across the x-axis. The yellow shading indicates the winter spawning seasons, and then the colors, the different colors, are indicative of when the fish was tagged, which season. So our first winter, we tagged a bunch of fish, all noted with the blue points. And you can see we saw some of those fish continue to return to Cox Ledge all the way through to 2023. We saw some fish kind of stay throughout the year. Others left the area and came back. But across the board, we see high residence to Cox Ledge, as well as a little bit to the broader region. And so now that South Fork is operational, we're looking to kind of replicate our work during the post-construction operation phase to really tease out if there are any changes to cod spawning dynamics. So we have 100 more tags to put out. We've put 5 of them out on the Orsted Revolution and Sunrise Fisheries Monitoring Trawl Survey in February 2024, but we have 95 more to get out, and we hope to leverage the New England Aquarium and Inspire receiver array, which is what Connor discussed, as well as potentially put out more receivers to really replicate our study. But yeah, stay tuned for that. In general, we saw cod spawning during the winter months on Cox Ledge, a lot of activity in those areas that I pointed out. We saw interannual spawning site fidelity, the fish coming back each year, as well as some broader residents. And again, we're looking for funding to kind of complete that post-construction phase, but hopefully I'll have more results to share about that soon. And I'd just like to give thanks to our fishing partners, Captain Greg Mataronis and Rick Bellavance, as well as all of the funders and people who have helped out on tagging trips. Thank you.
All right, well, good morning, everyone. I'd just like to start by thanking Chris and Greg for organizing this, and I'm happy to present some of our results after we've been conducting this project for 16 months, and a lot of my collaborators are on this stage, but we have a whole bunch of people. I'm not going to list them all. So this project was funded by Orsted to look at effects of the cable that comes in near Wainscott on the South Shore of Long Island, and we have several components. We have essentially a 16-month period prior to construction and operation of the cable, and then we're going to have a component after the cable is turned on and be able to evaluate whether or not we see any significant effects on fish movement. And in addition to construction and other activities, one of the main components of our project is to look at the electromagnetic fields produced by the cable that comes into shore, and whether or not that has the potential to change fish migratory patterns or local behavior, either attracting the fish to the cable or repelling them from the cable. And fish aren't all the same in terms of how they respond to electromagnetic fields. Some are much more sensitive, such as the elasmobranchs, the shark skates and rays, but all fish to some degree respond to electromagnetic fields. So we set this, this project up with this system shown here, which thankfully Chris showed a picture of where it was on Long Island, but it's on the South Shore of Long Island. And it has 3 components. It has 3 traditional gates along the South Shore, and this is actually contributes to a network we have across the New York Bight. But we have the west approach and the east approach and the cable approach. So if you look at the red dashed line, that's the cable path. So that middle approach is designed to really be right along the cable, and the west and east approach will look at fish behavior as they move into the area, and then we can look closer to the cable and compare those three components of the array. The 4x5 grid there is a fine scale positioning system, and that's what I'm going to focus on today. And in this position, all tagged fish we could 2D position, and fish that have also equipped with depth tags we can 3D position, which we're doing with some of our sharks and our striped bass. So that's the setup, and I'm going to talk about our results on the fine scale array today. And just to give you a little background, of course, we have to be able to track the behavior of fish. Our group has put out 1,400 plus tags across 23 species, and these are the partners on this project, but we also collaborate in a lot of data sharing networks, so there's more fish than that that have the potential to come through our system. In the first 16 months, we had 260 individuals across 17 species come through the fine scale array. So we're getting a pretty good coverage across a large number of species. And one of the ways that we judge the performance of this fine scale array is through three different measures of air. The first is something called the horizontal positioning air sensitivity. And this is a synthesized air because you don't know the exact position of the fish to compare its location to, but it gives you a relative measure of how well your array is doing in terms of positioning the fish. There's a second measure that we also look at, a horizontal positioning air that has this added M at the end of it, and it is not unitless like HPE. It is measured in meters. And that is based on sentinel tags that are inside our receivers. So inside that grid, every receiver has a tag that operates just like the tags do in the fish, sends out a ping, but now you have known locations. So you can actually position your receivers and get an idea how your array is positioning both the other receivers and likely how accurately or precise it's positioning fish that move through your system. And finally, it's not just space, it's also time that contributes to the error of your positions. And that's the root mean squared error, which is a detection time error, so that you're looking at this in time and space to come up with a total error on your estimates. Sorry for all that. Now we'll jump into actually seeing some of the results. Again, there were 17 species. I just picked 3 to show some results in this first section here. So those X's are the receiver locations zoomed in on the fine scale array. This is for summer flounder. We're very excited about these results. This is one of the first systems to be put out in this type of a marine environment, and we're getting highly precise movements and refined ability to track their movements through the system. And each of those different colors represents a different individual. So in some of our cases, we're able to look at these and sort of derive what kind of behavior they're doing, whether or not they're migrating through or foraging and so forth. Another example, clear-nosed skate. This one has more individuals in this first 16-month period, and I know this is a bit messy, but it's a lot of individuals moving through that we're able to estimate their positions on a very fine scale. So really this has worked out very well. And if you just imagine for a moment that there is an effect by an electromagnetic field, you could see this distribution of points shift, either closer to the cable or further apart. And so in the next part of the project, that's what we're going to be looking at to see whether we see those kinds of effects. And finally, for the last of these figures, this is Atlantic sturgeon. So Atlantic sturgeon, so far in this first 16 months, appear to be moving horizontally along the coast of Long Island in this area. So you can see their sort of migratory movements through the array. You will note that their dots and positions are further apart than the other 2 species. This is largely because sturgeon have more powerful tags, so we have a longer delay on them. So the number of pings going out has a greater time distance between them. So they're moving through and we're getting less dots per area, but compared to summer flounder and clear-nosed skate examples that have a higher ping rate. Okay, so now you've seen some of the examples. We're pretty happy with those. I want to to go over some of the error estimates and how our array is actually performing to be able to address this question, whether or not there is any effects on movement. And on the left there, I just show some of the top species and detections from sturgeon, black sea bass, clear-nosed skate, summer flounder, little skate. There's a total of about 54,000 detections in that first period. And over here, this figure shows 3 download periods, which represent those 16 months. So from download 1 to download 3, this shows HPE and RMSE. And basically, for understanding these figures, the green are very good positions, so very low error for these kinds of systems. And as you move to the red areas, they become less accurate. So we're actually very happy with these results. You can see most of our results are in the green across both for HPE and also RMSE. So our error estimates have been really performing well. A couple things I want to point out. If you look up at the red dots right there, the further you get away from the array, you start to see greater errors because they can't position with 3 or more receivers. Okay, so I briefly want to talk about receiver loss. This is an active fishing area, and we do lose quite a bit of receivers. I only have 2 minutes, so I'm going to go a little bit quickly here. But during this period here, we lost 5 receivers. And a loss on 25% does not appear to impact our ability to position fish very well, given our grid design. So you can see moving across here, that's the HPE time series on the top, RMSE on the bottom. Those are the 4 receivers that were lost, and then a 5th one. After this 5th one, those data are nonsensical because we were actually pulling receivers and downloading data during that period. We saw a slight increase in both of our error measures with a 25% loss. When you get greater losses than this, it does start to impact the system. But on this level, we feel pretty confident that our system is still able to position fish with relatively low error. Okay, so just to conclude, we feel that our findings so far suggest that our fine scale positioning system can effectively position fish, likely below a 1 meter resolution. And we do feel if there's a significant effect of the electromagnetic fields on fish movement, we're going to be able to detect it. And receiver loss remains a challenge, and it does, if it gets over 25%, start to impact performance of the array. And we did have a period last fall where we saw that. I think we had a 40% loss. Don't quote me on that exact number. But our next steps are to start looking at this data after the system's been powered up. So, thank you.
Good morning. I'm Brad. So we're all sitting in this room because there's very large scale and large anthropogenic changes along the coast, and we're trying to understand how is this going to affect the organisms that live in that marine environment. I'm going to be talking today about Sunrise Wind, which is a project that we're involved with, and we've talked a lot about kind of the impacts of the actual construction, the hard substrate offshore, the scour material, the impact on hydrodynamics. What I'm interested in, as Mike had talked about, is these cables that are running from these turbines onto shore. And this one is coming in at Smith Point in New York. And I'm really interested in the animals that are probably most sensitive to these magnetic and electric fields that are created out there, the elasmobranchs. So here is just a BOEM kind of survey of all the different organisms that are sensitive to these electromagnetic fields. For this project, we fixated on these 5 species. So we were interested in duskies, sandbars, smooth dogfish, sand tigers, and winter skate, with really the desire to evaluate the effects of the export cable on fine-scale changes in behavior and movement in the near-field environment around the cable, and assess these effects of EMF on behavior and movement of these targeted pre and post. That was our goal, our desire. To do this, we originally put out a 38 receiver fine scale array, like what Mike had just talked about. We did this with consulting much of the fishing community on the south shore of Long Island. They basically had a big input on where we put them at depth. So we moved them in, keeping out of that 55-foot depth range. You can see from that grid all of those lines of red near shore were where we had lost receivers the first year, primarily because of wave energy that broke the buoys off of the acoustic canisters, and so they fell over and got covered in sand. So we've gone down to now a 21 acoustic receiver array, but you'll notice close to shore those 3 green dots, and so we're really trying to get as close to land as possible, and those are a different kind of canister that we think is going to be more robust, and so we kind of split the difference. To date, we've basically tagged 67 duskies and 45 sand tigers, 51 sandbars, 67 smooth dogs, and 1 winter skate. I am also relying on tags put on by other people here, but we're putting more of an effort on our skates right now. Since the 2 years, we've caught approximately, over 300 different individual sharks. We've tagged over 200 of those sharks. When we bring I'm just going to make a mention, when we bring these animals to the side of the boat, not only do we put a tag into them, but we're going to do a workup of the size of the individual. We also take a fin clip for genetic, we take a muscle biopsy, we collect blood, and we're also taking cloacal swabs for fecal DNA. And I mention that because there might be some people in the room that are interested in that kind of data. This is just giving you a picture of where these animals have been tagged, where they actually hooked and lined them and then put the tags on the south shore of Long Island. What this is showing you is that since 2022, within our array, we've gotten over 4 million different detections, which is about over 1,100 unique individuals. What I'm showing you are only tags from our animals at this point. The ones that we've tagged. You can see that this is an early summer, early fall kind of presence, and then they move. But we're also using pressure tags, and so you can look at things like kind of where they are in the water column when they're spending their time around the array. And of course, we're interested in how that vertical distance might change when the power is turned on. So this is just some of the depth data from these 4 species within our array. Now, here is our positions of all of our animals. The 2 different colors are the 2 different years. Okay, so this is just giving you, we've got 9,000 different positions across this array in the last 2 years, and the color just gives you the year that it was done. But, which I just want to say, we've got a lot of positions across the array. That's great. But what's really interesting, and looking at it a little differently than Mike showed you individual movement, which is, of course, something we can do and is great interest. But we're also doing things like dynamic Brownian bridge movement models. And this is what I'm showing you here, kind of where this is just showing you the sand tiger. So we had 9,000 positions of sand tigers. We've lumped them all together. We've looked at kind of how they're spending their time within that array. But, of course, we can do that with the other species. So here's just what we have for the sandbars and for the duskies. So I feel pretty happy with kind of the number of our, because I haven't even put Keith's individuals or Mike's individuals into the array to look at all the other elasmobranchs of these different 5 species that have been tagged. This is just showing you what we've put out. So I feel pretty confident that the pre-switch on, we have a pretty good idea of how these animals are moving across this space. But I also want to, kind of talk about the bleed over effect. And this is one of the, this project is important for the task that we are trying to address. But our animals have been detected on, I think it's 28 other arrays across the East Coast. They've been detected from Canada down to Florida. So these animals are showing up on other arrays other places, and just like Keith and Mike's animals are helping me, these animals that we're tagging will be of value to other people in other regions. And I just want to say thank you. This is a colossal effort to tag. We're going to tag 100 individuals of these 5 different species, although we have a lot of winter skates to tag yet. But that's our goal.
All right. Hi, everyone. My name is Matthew Sclafani. I'm with Cornell Cooperative Extension, and I'm going to talk to you today about horseshoe crabs and lobsters around the sunrise array that Brad just described. I was told I have far too many slides for this 10-minute presentation, so I'm going to probably speak a little bit quick. But overall, I just want to say that the main goals of our project are to use acoustic telemetry methods to study the behaviors and the movements of these 2 benthic species. And we're going to hopefully use the behavioral metrics that we get from the acoustic telemetry data to say something pre and post construction of this export cable and determine if there's any potential effects of EMF on them as well. So we decided to study horseshoe crab and lobster for a number of reasons. First, they're important fisheries in New York State. These species are also benthic, so they're going to be more closely associated with the export cable and the potential EMF fields from that. So we hope that would be something that you can detect as well if there is a signal. And we are also seeing a species that's migratory. Both horseshoe crab and lobster do migrate and move around considerable amounts. We're going to be potentially seeing them in the sunrise, not only in the sunrise array, but also in the South Fork array that Mike spoke about, and potentially other acoustic arrays. Lobster have also been shown to have some potential effects from EMF, as has been shown in other studies. And horseshoe crabs have also been listed as a priority species of interest by the Bureau of Ocean Energy Management. So here's a plot of the positional array. This is something that Brad has already showed. This is the sunrise array. It's off the south shore of Long Island, pretty close to Smiths Point County Park. The dots are the individual acoustic receivers, and the red line going through is the proposed export cable run. So our goal was to just simply tag annually up to 75 horseshoe crabs and 50 lobster. We are using Innovasi tags. These tags are attached onto the external, their carapace and their shell, and that's just done by simply using some adhesive, basically crazy glue and some Velcro. These methods are proven to last quite long. We've had tag returns up to 3, 4, 5 years of individuals on other studies. So we're confident in this method, but we also use a double tag approach just in case. So we use button tags from the U.S. Fish and Wildlife Service and Floyd tags on the lobster. So far to date, we've tagged over the last 2 years 150 horseshoe crabs, and we've tagged over 100 lobster. These individuals, the horseshoe crabs, were captured here by hand and using boat in Mauritius Bay, which is the embayment behind the South Fork Wind. And what we do is we would collect them, measure different metrics on them, including size, sex, carapace condition, and then we would tag them and then place them directly into the array. We'd go back out and place them into the center of the array. For lobster, we did a very similar approach. However, we worked with a commercial lobsterman who helped us to collect these using baited traps. They were collected off of the artificial reefs from the New York State DEC Artificial Reef Program. These tagged lobsters were also measured for their end sex and also looked for their condition of their shell. Once they were tagged with the V13 tags, they were then released into the positional array. So real quick, just so you see the size of the animals that we're working with, we have the typical sexual size dimorphism for the horseshoe crab, where the female prosomal width is considerably larger than the males. And in inverse for the lobster, we have the males being slightly larger than the females. But overall, we have a pretty good size range to work with and to make some potential inferences with respect to covariates in these models. So as with the other presenters, we actually have had incredibly great detection success in this positional array. For the horseshoe crabs, we had over 1.3 million detections just on them alone. So we're getting really high number of positional estimates that we can derive from those from these detections. On average, is about 8,934 per individual detections. But the really important number is the mean number of days that they're spending within the positional array. And it looks like on average right now, we're getting about 11.6 days of residency time by the horseshoe crabs. So, this is all very preliminary data. The cable has not been constructed yet, so this is the pre-construction data. And I just wanted to also plot some of the trajectories and some of the overall positions of these horseshoe crabs that you calculate. And when you filter the data, you get about 128 individual horseshoe crabs that we could work with out of that sample that we released. So pretty high yield. And you can see here, when they're released in this array, they're using a great deal of the spatial habitat within the actual positional array, and then they eventually move outside of the array. So the line that's going through, I just want to kind of focus in on this, is that's the export cable, the proposed line. And what we see here is I'm just going to hone in on just an individual track of one female that stayed within the array for 6 days. The green circle there you can see is the release point, and that was on September 14th. And then the black circles are the actual position of the track of this individual moving through time over the 6 days. And then the blue circles, the blue line is actually a predicted correlated random walk that we'd use to see if we can model the actual movement behavior of this animal, and it does a very good fit. And what's important here, though, you can see that the animal actually traversed this cable twice in this run. Then just another individual. Here's a male that actually was a much shorter time frame, but it too also crossed over the proposed cable. And so we might be able to say something about its export. It actually popped out of the positional array and then jumped back in March, so about a couple of months later. So we have to see what happened to it since then. But you're able to track these positions. And again, we're getting good model correspondence with the correlated random walk for estimating these movements. When we took a quick look at the size distribution and the total distance traveled of these horseshoe crabs, we didn't see any relationship between them. Likewise, we did not see any sexual sex difference between the distance traveled between the males and females. So both are using the space equally, and that's going to factor into our analysis as well. For the lobsters, we even did better. We got about 2.24 million detections, so we were really pleased with that. That's a heck of a lot of data to work with. We have a mean of about 22,000 detections, and they have a much higher residency time of about 41 days that they're staying within this positional array. So we're pretty excited about the potential to see what we can get from this data. So, when you look at all that, it's no surprise when you spend more time in the array, you get a lot more habitat use, and we're seeing really good coverage, good spatial coverage by these by the positional arrays, and we're really pleased with that. When you actually look at it by sex and you do the Rorschach test, you can see here that the males have a slightly more expansive range. They tend to be a little more exploratory in their nature and using more of this habitat relative to the females, but overall, both are using the full detection, the full positional array to explore. Now, when we just take a quick look at one individual here, again, releasing at the green point, this individual stayed in the array for about 3 months of time. And you can see here that it started out rather exploratory, and then it traversed the cable several times until it wound up into an area of attraction, which is denoted by that red circle. And it kind of spends some time, it's probably a habitat that it liked, and had more of a foraging type of behavior. So we can start to glean some differences about maybe some behavioral states that are going on from this type of data. Again, when we checked for any relationship between carapace size and total distance traveled, we didn't see any relationship, and there was a slightly increased use of total distance by the males, shown here in the blue, relative to the females in the red. When we look at day versus night movements, overall, just the total distance traveled, again, these are very simple metrics that we can run quickly. We did see that there was actually a slight increase of movement in the nighttime, so this is kind of consistent with some of the lobster biology. And then when you look at that by sex, there's also a slight increase in movement by the males relative to the females as well. So The future of this data is actually very exciting for us because we're going to pretty much be able to dig into a much deeper analysis. Again, this is very preliminary data, but we can use things like home range estimators and kernel density estimators to talk about those core use areas, and that's what we'll do with those other plots. But what I'm pretty excited about here, actually most excited, is the potential for us to use the hidden Markov models, which is a subset of state space models, and that enables you to kind of incorporate other covariates such as time, tide, sex, and all kinds of other factors to see how the animals behave. Just to get a quick look at that, this is sort of the turning radius of an animal. If you use that with the step lengths, you can actually make some differences about the hidden state or the actual state of the animal, which might be like a long step length and a very, very little turning radius. You can say that that's more of an exploratory type of state, and that would be exhibited by the blue over here. And then if you have a lot of turning that's going on by the animal, very tortuous in nature, compared to what you would have for, it would be a second state, which might be more encamped or foraging type of state. And you can see this is a 2-day trajectory of an individual that allows you to talk about the transition from one behavioral state to the other. So in short, I'm already over on time. We have just pretty excited that the telemetry data that we've got so far is yielding very high resolution spatial positions in both of these species. The horseshoe crabs have a little bit more limited time spent in the array. However, they are still going to be able to potentially give us some very good insights into any effects of EMF. What's kind of neat, too, is the tags can last up to 5 years, maybe longer. So we hope to see subsequent movement. We've actually seen 13 of our horseshoe crabs tagged in 2022 pop back in 2023. So, we're seeing multiple occurrence of use of this habitat. Lobsters are really good in that they have an increased temporal and spatial use of the array. They utilize fully that whole area, and it looks like they'll be able to allow us to do some very detailed analysis with hidden Markov models as well. So I'll leave it at that. Thanks.
Good afternoon, everyone. Save the best for last, obviously. I'm Keith Dunton. I'm from Monmouth University. If you don't know, Monmouth's are about right there, which puts us kind of smack dab in the middle of all this offshore wind. Today I'm going to be talking about kind of this area and region and offshore wind lease sites. I like to put faces to names, so this is a little bit of a snapshot. Josh Kohut showed this morning, a snapshot of our research team that all kind of help out in various ways and forms. So acoustic telemetry is a huge tool for non-extractive regional monitoring, really important for some of the species that I study, like sturgeon and sharks, because we can tag one animal and get 10 years of information from it. So we'll be focusing in on this, and this is a combination of a variety of different projects. But this area and region is really important because it's kind of a known migratory highway for a lot of species already, particularly the anadromous species, where we have 2 big estuaries, the Hudson and Delaware Bay. They're moving back and forth, as well as coastal sharks that are moving northward during the spring and then southward again during the fall. We also have a really big offshore inshore component of fisheries like summer flounder and some sea bass and stuff. They're moving inshore during certain months and then offshore during other months. So it's a really dynamic region for lots of different fish species. So the overall goal or arching goal that we're aiming to look at is just examining the effects of offshore wind on the distribution, connectivity, and migratory behavior on lots of different fish species. Particularly this region, which is funded through the New Jersey DEP RMI program, takes an approach looking at a variety of offshore wind lease areas, as well as we are working up in Equinor wind through Inspire and Equinor for Empire wind and examining that region. I'm mostly going to be talking about the RMI project today, mostly because we just finished downloading our receivers last week. So we are in the pre-construction phase of monitoring, so we're aiming for about 2 years of monitoring on those sites, but we're hoping to continue the surveys for post-construction as well. Along the New Jersey coast, we have many different species to pick. It's hard to pick just one. The state surveys identify up to 214 finfish and important invertebrates to look at. Most of these species are commercially or recreationally important or ecologically significant, like sturgeon or prohibited sharks. So we took kind of a snapshot approach, multi-species. We're going to tag a little bit of everything in all of our projects to kind of get more of a holistic view of some of the changes that might be happening. So we're using a variety of different tags, from the smallest V9 up to the largest V16 on a lot of the species. We're also using the acoustic release Nova C receivers with a release recoverable mooring system, which is this here, and that's a receiver with a canister of rope, so that when we're ready to recover the units, we can send the receiver up, trailing the rope. We pull everything up to the surface. I'll have a slide on why that's important in a minute. And then we also try to equip things with snapshots of turbidity sensors, DO sensors, salinity sensors, anything we can do to try to get environmental data on a lot of the lease areas that we have. The need to be ropeless is important in our region. We have a lot of humpback whales that are feeding a lot of the times. So these systems sit about 5 feet off the surface, and they're really, really, in my mind, compact to some of our older systems that we have. And we have them ropeless as well for gear interactions, to try to reduce gear interactions or to try to minimize impacts to commercial fishermen. It makes it a little bit complicated for us as scientists because these systems are a little bit wider and bulky as we're trying to deploy them offshore. To date, amongst all of our offshore wind projects, we've deployed 136 receivers across a variety of different wind sites with Equinor or Empire Wind up here, and then these are our RMI program. The RMI program, we've deployed 82 receivers in what we're calling a variety of different sites: the North Cable Landing, the South Cable Landing, inshore lease areas, and then offshore lease areas. We got a lot more density of receivers here because those lease areas are further along in development than the offshore lease areas. So we're hoping to get some baseline data out here on a lot of the fish that we have. Within our RMI program, we're focusing on trying to be as robust as we can. So we have all the receivers in the lease areas themselves, but then we're trying to monitor the cable landings specifically, so that's why we have them bracketed, so we can look at residency within those cable landings, movements across the cable landings, how fish might respond or change to the cable and cable depths as they're going, as well as a gradient approach within the most advanced wind farms right now. So this would be in lease, inshore of lease, and then offshore of lease. So how do the fish respond when turbines are in the water, and maybe they change their behavior? So we're hoping to get a nice snapshot of that. We take as cooperative approach as we can, working with commercial fishermen to deploy our gear, as well as tagging fish. This is the Berkeley Striper Club. They're the most amazing group that I work with. They even make ourselves a little sign when we're out, and they like to feed us and take us all along the beach to tag striped bass. Colleen still has to get out with us when we do that. But we work with all different groups and recreational anglers, as well as, that's Captain Kevin work. I had to take that picture because I find it funny that the commercial fisherman broke out his little rod and reel to try to catch us striped bass when we were out there. He was unsuccessful. So the status to date, we have deployed across all of the RMI program and Equinor. We've tagged 350 tags so far. We have many, many more. I feel like that's the common theme about everyone. We're about halfway there on our deployed tags. This year we were largely focusing on sturgeon because that's a really important species. So we were pretty successful at tagging a little over 100 this year. but we're tagging a little bit of everything. When we combined our offshore wind tags with our other tags, we have a little over 560 tagged. The flagpole's in the way. But we're focused on everything from a variety of different sharks and rays and skates to all sorts of fish like blackfish, sea bass, flounder, and such. 2 minutes, holy cow. So far to date, we've detected 1,500 unique animals. Of those unique animals, we've been able to identify roughly about 800 of them. But we can see a majority of the detections are coming inshore here with a lot less offshore. Of those animals, 22 species detected, and this is largely our tag group, Stony Brook's tag group, and some other regional researchers. Does not include Jeff Kneebone's tags, which are probably pretty important. But we've detected everything from white sharks to all sorts of fish. The most abundant fish, though, are sturgeon, striped bass, sandbar sharks, and dusky sharks. We do have a large amount of unknowns. We are and will submit this to the regional networks in order to identify those fish and tags. Looking from inshore to offshore, we can see differences amongst the different tagging groups and regions. Inshore, we see a lot more of our anadromous species, sturgeon, striped bass, alewife, and then as we go progressively offshore, there are less receivers, but in our offshore arrays, we tend to see mostly the elasmobranchs, the shark species that we have here. Up next, we have lots of recovery and download to do, but we have lots of tagging efforts to go through with our HMS. I'm going to end there since I'm out of time, and I think the challenges that I describe will be lots. So we'll end there. I feel like I could spend an hour on challenges.
All right, just a quick reminder, if you want to ask a question, please put it in the chat. We got a lot of questions coming in. If there's one that you like and you want to see answered, please give that an upvote so that it goes to the top of the list. I'll start with a question that's really open to anyone up here. Thinking at a broader scale, what do you guys see as the biggest challenges that remain when we think about a regional approach to telemetry monitoring? And then conversely, what progress have you guys seen over the last 2 years since the last State of the Science workshop? Easy one.
Well, I had a lot of list of challenges. One of my major challenges as far as regionally goes is the developers not talking to each other or making it hard for us to data share or contribute towards the bigger regional data sharing groups like the MADOS network. If you don't know, the MADOS network is a repository where we can submit all our tags and it helps us distribute and able to figure it out. That's been my biggest challenge with some of this stuff. I don't know if it's the same across the board.
Don't be shy.
I think one of the big challenges that we had was, just trying to, besides if you have different tags types that are detected by different companies, that's definitely something that they're improving upon, I believe. But getting some gear interactions with the active fisheries have been challenging. Not so much on Sunrise, but South Fork has had some issues. I know Mike mentioned that. So we need to work better with those, with the fisheries, I believe, and the commercial fisheries to make that less of an issue. And I think there's ways around that we haven't fully explored, but we can definitely do a better job at that. And I think that would be very helpful overall.
I was just going to add on to what Matt said. So for the Sunrise, right, that fine scale array in 2 years has lost 26 receivers, right? That's basically because the first line was too shallow. We mitigated that. But then the last 2 years, there's been cold slivers of water near shore, and it's allowed the squid to get in very shallow. So we've got draggers that are dragging in 40 feet of water, which is not normally where they would go, but that's because that's where the squid is. And so it was an unforeseen interaction because they said as long as you stayed, kind of like 50 feet or more, you'll be out of our way. But then that changed because that's where the money is.
Yeah, I'll add to that, I think across all the projects we've lost receivers, and, we've been out doing this since, I guess, 2010. Is that right, Keith? Yeah, I think that's when the sturgeon arrays went out from the tip of Long Island down to Atlantic City. And we've always had losses, and in some cases we can put systems in areas where there's less fishing activity. But for instance, if you're monitoring the cable, the effects of the electromagnetic field from the cable, there's only one, you don't have a lot of options of where you can put your array. You have to put it along the cable. And as Brad mentioned, the squid fishery has been inshore the last few years, and that's caused losses. We changed our design in consultation with some of the captains to come up with a design that if they do catch it, it's less destructive to their gear. And we've actually had a lot of success with that in terms of we get a lot of calls from captains. I got one of your receiver setups, come pick it up, and then we go put it back out. And so we've gotten quite a bit of success in that, but we still have some pretty significant losses during some periods where we don't know what happened to the receivers. And those periods I mentioned last fall was one of them. And I don't know exactly how we improve because there's been communication, but it's a difficult thing because people are out fishing as part of their business, and the receivers in some cases are in places where that activity does occur.
Yeah.
Okay. One thing of note too, just to keep in mind, is that many times these guys will catch one of the receivers and they'll drag it, but they won't take it away. And so because your array is so dependent on positioning, what that means is that you have to double or maybe triple position your animals based on the downloads. You have to go through the day. So it's greatly increased the effort to try to position the animals because you may have to break it up into multiple sections.
I'll just add one thing, Greg, too, is that we're talking about acoustic telemetry, but these wind lease areas are very complicated animals in that now we also have benthic surveys, we got clam surveys, we got trawl surveys, we got NOAA scientific surveys, we've got state surveys, we got hydro surveys, and now like everything is all being coordinated or trying to be coordinated, and I'm on like 10 WhatsApp communication things, and it's getting a little bit complicated with some of the logistics of things. Sure. That's why I like that Rose's in fish app or coordination app or something.
Yeah. I like to just add, I think the squid are attracted to the positional array and like it.
Yeah, we haven't figured out how to get a tag in the squid yet. But I did want to mention before we go to another question, Keith's first point about data sharing. I think one of the challenges over the last 2 years, but really over the last 20 years, is that there's different players with different incentives, and Keith mentioned different developers have different policies about data sharing. But there's also, academics versus non-academics that are doing the research at different incentives. Academics are trying to answer the question that the project is addressing, but they're also trying to train graduate students and have publications, and so they have a different incentive structure than a private contractor or consulting group doing putting an array out. So it's very complicated, not just amongst the different developers and other funders, but also amongst the groups that are doing the research, which I think makes it challenging to come up with something that works for everyone.
Thanks, Mike. And I think that actually answers one of the questions we got here, but I was wondering if some of the other panelists might want to expand on that. And the question is, what is the justification that we get from some entities, whether it's developers or academic institutions or other groups, for not participating in these larger data sharing networks. So I think, Mike, you spoke to that maybe from an academic perspective, but I don't know if anyone else has anything they'd like to add there. Real softball questions here.
Yeah, I think Mike made a good point about it, but also with what we're doing for offshore wind, we do have to do things on a relevant time scale. You can't hold on to data for 100 years if we're trying to figure out if we're having impacts on something like endangered sturgeon. So I think there does have to be a timely manner in trying to analyzing this stuff, but also being aware of protecting students and graduate students and training workforce.
Thanks. Anyone else? We got plenty of other questions. So, all right. I guess kind of relatedly, what are your perspectives around how some of our federal partners could help better facilitate regional monitoring? And maybe that comes through improved guidelines or requirements, or are there any other avenues that you guys see that would help kind of get us all more on the same page when it comes to how we go about sharing and regionalizing telemetry?
Yeah, guidelines could help. As of right now, there's no written guidelines on acoustic telemetry, or making requirements in permitting requirements through whether that's through BOEM or through some of the permitting stuff.
I think it would be good to have them, but I also think that they could very easily lead to more problems. And again, there's different incentives for the different groups that are involved. And so I don't know if you're going to come up with one model that's going to meet all of those objectives. And it even comes down to your institution. Here we're an academic institution, and the kinds of research we're allowed to do that the university is going to agree to do, it does involve certain requirements on data ownership and publication rights. I mean, that's just the way most universities operate.
Yeah, I think Dave said it best. He wrote his plan, then he had to stick to it. Terrible.
So
it always looks good on paper.
Yeah.
All right. I guess this is open to the whole panel. Do you guys have any information on post-release mortality? And if you've seen any post-release mortality, how you're handling that and what it may mean for the analysis?
So, I mean, I'll comment on that. I mean, we see very little post-release mortality. I mean, I just think about sturgeon. We see, I don't know what the number is, but for a while we were seeing, I think, over 90% of our sturgeon every year. So I think it's fairly low, and I think it's quite. I don't have an exact estimate. We never estimated it, but we do keep track of what we tag and what we detect.
Yeah, for the horseshoe crab and for the lobster tagging, we've had very low mortality from what we can tell. In fact, some of our lobsters have migrated back to the artificial reef and been captured by recreational fishermen and some commercial fishermen. So, that was the benefit. They do, the lobsters, they will molt, and so they'll shed that tag after about a year. The horseshoe crabs have a terminal molt, so they won't shed it. So we see, 5, 6, 7 years sometimes of, detection. So I think the mortality rate's pretty low. If anything, it would be from either a stranding or a commercial harvest, potentially.
I'd like to add, I do have the problem of some of my species being caught and eaten. Sea bass and tog, because we fish for them on structure and reefs, and it's like you put a tag out, it immediately gets recaptured. I'm like, wait a minute, I know that was undersized.
Go for it.
I would like to add, I cannot speak to Jeff Kneebone and the work they've done in HMS, but discard mortality, there was a recent push in research over the past several decades, and it is largely unknown for many species, so it is something to constantly evaluate, especially some of our smaller species. HMS, for instance, can recover depending on the fight time and the capture and handling conditions, but smaller fish, especially those that are physocyclistic, which have a swim bladder that will come out of the mouth when they come up from pressure too fast, they need potentially, best practices to reduce that. So it's something to always be in consideration.
For our particular project, we've been working with a marine veterinarian we've been collecting blood, so they've been looking at kind of stress hormones to just make sure that the time that we have them along the side of the boat isn't leading to any overt stress. There's a high desire for the person putting the tag in to make sure that that animal leaves well financially. So effort is taken to make sure that we reduce that. Although I was going to mention that Mike Frisk did tag a black sea bass that ended up in Canada from a seal.
Anyone else? Good. All right, I guess I have a question that is probably directed at the folks who have the nearest shore arrays along the export cable routes, but when we get to the post-construction analyses, how do you plan on integrating things like EMF levels and other covariates into the analyses?
Well, I don't think we're quite at that stage yet, but we are going to try to look at that and look at the when the power is going out, how much power is going out, and try to correlate that to the movements. But our design was set up to look at a pre, during, and after. So pre-construction, during construction, and after the construction. And we haven't started those analyses, so I don't think I'm going to talk about specific models, but we are going to try to incorporate those covariates into our analysis.
Anyone else want to expand on that further?
We're not specifically measuring EMF levels in our study, but it is something we'd be open to if there was the availability of, the industry to help with that. However, we can, if we use the Markov models, we're able to incorporate those, at least as a presence-absence type of thing, to see before and after effects of an EMF on behavioral state switching or any of the velocity changes or any types of distributional shifts that we might see that would fall out of that type of modeling pretty easily.
We're cranking through these questions. All right, getting back to this idea of regional networks. I guess, can you guys talk a little bit about the benefits of being a participant in the network, but also what some of the downsides could be, or maybe the unintended consequences of sharing data more broadly? And I think there's probably a lot of experience here to speak to that. So I wasn't looking right at Mike.
Yeah, I'll say the benefits is this research is expensive, timely, extremely labor intensive. A lot of the species we're tagging are conservation need, HMS, and sturgeon. So I feel like, we put a lot of effort, time, and money into it that, looking at regionally what's happening off New Jersey, these sturgeon could be hitting like all of the lease sites all the way up to Massachusetts. So it's more thinking about the combined effects overall. A lot of the sharks are moving through all of these systems. So data sharing, I think, or regional sharing for that, and I think is very important when we're looking at like population level things.
And even for the question of the array, right? I mean, I have 1,100 animals that have gone through the array, of which I know about 300 of them, right? And so being able to position those other animals is just going to make the study that much more robust.
Yeah. So I think data sharing and being able to combine, researchers up and down the East Coast is vital to the science that's coming out of this. And you can imagine if you have 20 people that you have to communicate with data sharing. So you have the battles and these networks, but using that data, so you might know that you have somebody's data, but you still have to communicate with them with the use. And over the years, I've seen that change quite a bit. And I think one of the biggest challenges is just time. So if you have 30 different researchers that from Florida to Canada that you're interested in doing something with their animals, or they want to use your animals, that's 30 conversations. I mean, it's just overwhelming to try to do that. And what we've started doing more recently is just basically if we use a single data from you, you've been invited to the paper or whatever, and vice versa. And because reducing these amounts of conversations is almost a necessary thing. I mean, it sounds like a strange complaint, but having 30 negotiations between 30 groups for every project is just, it's crazy. It's overwhelming. And that's a challenge that has been with telemetry from the beginning, and I don't think it's going to go away. But obviously sharing data has a huge benefit to the private institutions and the public institutions that are putting money into these projects.
And I just going to add, I agree fully with what Mike was saying. And we've benefited from other studies from people telling us that they had our tag horseshoe crabs, and it gave us estimates of survival that we would not have captured otherwise outside of our array. So you really benefit. It just strengthens the overall integrity of a study in unexpected ways. I joked with Dave years ago that it's the gift that keeps giving when you keep getting data in. When people find your receivers that have been lost, or it really helps when you share data, obviously. And if there's ways to make that happen the way we're doing, that's good.
Yes, and maybe for the realm of offshore wind, maybe we can eliminate some of those conversations, Mike, by just having like an offshore wind consortium where all of the offshore wind developers and people working in it just pool our resources overall. That's just my opinion.
All right. I guess no one's got a crystal ball, but I mean, to me, the technology has come a long way in the last decade or so, and I only see improvements happening. So do you guys foresee any improvements in technology that are going to even increase our understanding further when it comes to impacts of offshore wind?
I think just across the board, more powerful tags with higher detection ranges, tags at smaller sizes so that we can capture information about, someone mentioned squid. We don't know how to tag squid yet. Perhaps a smaller tag could be beneficial there. Fish at different life history stages. The technology's come a far way, but I think there's a lot more we can do.
Yeah, I was going to say, I think Innovasi is about to release their newest line, which gives you 80% more detections and a bigger range, which I feel like that kind of messes things up. Because we have so much other invested in the other technology, but offshore, when they're out there, that's a better realm.
Yeah, and I'd like to just advocate for the other side of that story, which is while they're advancing technology tremendously and you're getting much more high-resolution data, that's exactly what you get, a lot of high-resolution data. So I'm advocating for the analytic tools to improve and the data handling as well. That's a big need.
I'd like to see if the Novasi's listening, that for the receivers to come up automatically and reel themselves back in. Airlift it, send all the things, and make it so much easier.
I think Matt raised an issue. We're collecting this data now. We have a game plan for how we're going to analyze the data, but I think it would be really good to have kind of a group of people who are doing all the similar things that we could get in a room and talk about how's the best way for us to analyze this data so that we're making sure that it's all kind of apples to apples when we make our comparisons.
Well, and also, okay.
No, I was just going to add that while it's not necessarily something that has been developed in the last 2 years, but the depth and acceleration tags, I think for these projects, particularly if you're looking at effects of electromagnetic fields, things like that, are going to be really useful. And the tags are a lot more expensive, so you're balancing sample size versus what you get off of each tag. And we've started in our striped bass deploying acceleration and depth tags, and so we haven't analyzed them yet, but I think there's a potential for that to show some interesting things too. Not just with the electromagnetic fields, but also how striped bass are changing their position in the water column and various things up and down the East Coast as they go through different habitat types.
I was going to just add that we also need to think about and have data preservation in mind, because when like Mike retires and his laptop goes from his office back to home, we don't want to lose all that data that we have here. That's where like a repository or data keeper would be really important. These offshore wind is going to have lifespans potentially greater than our careers.
All right. Some of you touched on this briefly in your presentations, but could we just quickly go down the line and talk about what the plans are for like study duration for each of these studies? Because I think that's been an active topic of conversation of like how long do we need to monitor for and how long should these projects go?
For the Sunrise Wind, it was 2 years before construction, during construction, and 2 years after construction.
Our current projects are currently funded only on 2-year increments. How long I think the project should go for? 100 years.
Yeah, I'm working with both Mike and Brad on their arrays, and it's 2 years pre, 1 during construction, 2 years post. Looking at my species that I'm working with of interest, we're getting pretty large sample sizes and considerable data. So if we can characterize that before construction well for the behaviors, and then at least a couple of years afterwards. But I would definitely advocate for longer time horizon in terms of because some of these tags are going to last 5-6 years after we tag them. So you might as well get as much out of that as you can. It's not inexpensive.
Yeah, we had 4 years of pre-construction baseline data, and now that South Fork is in operation, I would love another 4 years of post-construction, but we'll see how that comes together.
I got one. I cannot speak to Jeff and Brian's initial plans, but they do have funding through 2026, and again, echoing the rest of the group, having longer data, tagging newer individuals to make up that variability to understand the population better is essential.
Yeah, so people have already mentioned the South Fork Wind Project, but it was 2 years before, I think 1 year during construction and 2 years after. Obviously, having a little longer data is always beneficial, but that's what the current plan is.
Great, thanks. I think we got time for one more question, and it comes from Colleen. I guess we've really been focused today on offshore wind, right? And that's obvious, but what do you guys think about of some of the ways that we can use some of this data to better inform stock assessment? I think we've all got a desire to improve fisheries management and resource management, and so what are some of the overlaps there and how we could get more value out of this information we're collecting?
Yeah, so there's a lot of ways that we haven't talked about this kind of research, but we have active research estimating mortality in sturgeon, and we can actually get mortality down to a weekly level in sturgeon. We also have been doing a lot of work on winter flounder, and we've looked at contingent structure and migratory dynamics in terms of looking at residents and migrants, which we've verified with various acoustic telemetry stuff over the years, and that's actually not integrated into the stock assessment for that species. And there's more examples, but this information has a lot of direct a lot of importance to stock assessment. I just mentioned a couple there.
This isn't really directed to stock assessment, but I have a very intelligent PhD student who went to the state of New York and said, they're investing $1.7 million in acoustic tags. It would not take that much for you to put acoustic arrays on all of your offshore artificial reefs. And they were like, yeah, we should do that. And so, I think that leveraging this to understand things like habitat use in the state is really good.
Yeah, and I just want to say these projects are contributing to, across all these species, to things that can be directly lead to stock assessments, and I think that's actually an area that we should be focusing on. I mean, these other areas we talked about are important, but with the changes to the survey and other things, there's information that can be gleaned on stock structure, mortality, movement, all kinds of things that are going to be enhanced by the sheer number of tags that are out and the number of receivers.
I'll just add to that, especially for Atlantic cod, we just moved from 2 stock units to 4, and a lot of that was based on tagging work and genetics, and acoustic telemetry really informed the stock boundaries, which have resulted in a change to our stock assessment structure.
Yeah, and, it may not be, again, directly related to stock assessment, but you do learn a lot about habitat use from these, which Brad was mentioning. But you also get survival estimates. So if you have long enough time series on your tag and you're getting a significant, return rate, you can basically get at a marker capture and get at better estimates of mortality survival. So that could probably feed into some sort of stock assessment as well. But you're going to be able to see, I think, a lot about migration and movement as well that can potentially feed into habitat use and key areas to protect as well.
Great. I think we are out of time. Thank you guys all very much for the presentations and the discussion. Hold on.
We have a great one-pager. RWSC and Rosa have some data sharing guidance, so come up on the way out and take them out.
Great.
Thank you.
Thanks, guys. Nice job. There's a few questions in the chat that are directed. You've got a couple for you. Need an app for everything. Can't even go to a conference.